Welcome to Section 2 of Cellular Biology. In this section, we'll be discussing the structure of the cell as well as some basic functions. Let's get started. This is a figure showing the components of the cell which can be found in Section 2 of Cellular Biology. Hopefully between undergrad and the MCAT, you already have a solid understanding of much of this information, so hopefully it'll just be a review. Notice from the figure that there are many organelles, such as the nucleus, the rough endoplasmic reticulum, and others. This is a table of many important structures of a cell which can be found in Section 2 of Cellular Biology. Notice that the table also includes some of the key functions, as well as clinically relevant information shown in the notes column right here. Let's go through it together now. As you hopefully know by this point, the nucleus is an organelle found in eukaryotic organisms that stores DNA. You can see the nucleus right here. The rough endoplasmic reticulum, or RER, is a network of membranes in the cell that's continuous with the nuclear membrane. It's said to be rough because it's bound to ribosomes. The rough ER is a site of protein synthesis. More specifically, it is the site of synthesis of proteins that will be exported to the outside of the cell. An example of this would be a peptide hormone, like insulin. On the other hand, proteins that will be used inside the cell are generally synthesized in the cytosol, not in the rough ER. You should also know that the rough ER in neurons is referred to as Nissl bodies. You can see the rough endoplasmic reticulum right here. Notice the membrane-bound ribosomes right here. The smooth endoplasmic reticulum is similar to the rough ER, but it lacks bound ribosomes. It's the site of lipid and steroid synthesis. A good example of this is a steroid hormone like testosterone. You can see the smooth endoplasmic reticulum right here. Notice that there are no bound ribosomes to this part of the endoplasmic reticulum. Next, we'll discuss the Golgi apparatus, which you can see right here. The Golgi apparatus looks similar to the endoplasmic reticulum. It's a complex of folded membranes and vesicles. It's responsible for modifying proteins and transports these proteins to their target location. One way the Golgi apparatus does this is through the phosphorylation of mannose residues on proteins. Normally, the rough ER adds some sugar molecules to proteins, and once the protein enters the Golgi apparatus, these sugar molecules are modified. There are several examples of these types of modifications, including N-oligosaccharide modifications, O-oligosaccharide modifications, and mannose 6-phosphate modifications. The most important to be familiar with, however, is the phosphorylation of mannose. When this type of modification occurs to a protein, it directs the protein to the lysosomes. The reason this is important to remember is because when this process is disrupted, it results in a clinical disease known as eye cell disease. Eye cell disease is a lysosomal storage disorder caused by a defect in N-acetylglucosamanyl 1-phosphotransferase. As we just discussed, this is the enzyme responsible for adding phosphate groups to mannose sugar molecules on proteins. When this is defective, the proteins that should go to the lysosomes are misdirected and end up being secreted in the extracellular space. This disorder commonly presents as an infant who has coarse facial features, failure to thrive, cognitive impairment, and corneal clouding. A buzzword that's commonly seen on exams is coarse facial features, so definitely remember that one. Okay, before we move on to the next structure, this would be a good time to discuss cell trafficking. This is a figure of cell trafficking which can be found in Section 2 of Cellular Biology. Notice from the figure that the Golgi apparatus can send vesicles to different locations. You can see these little vesicles right here. In addition to directing proteins to lysosomes, the Golgi apparatus also directs vesicles to and from the endoplasmic reticulum as well as the plasma membrane. Vesicles can be coated with various proteins, and there are 3 that you need to be familiar with for step 1. These include clathrin, COP1, and COP2. Vesicles coated in clathrin are found in vesicles going between the Golgi apparatus and the plasma membrane, so right here. These can also go to the endosome, which you can see right here. COP1 coated vesicles are found in vesicles going from the Golgi apparatus to the endoplasmic reticulum, so this way. COP2 coated vesicles are found in vesicles going from the endoplasmic reticulum to the Golgi apparatus, so this way. Okay, continuing along, processing bodies, or P-bodies, are regions in the cell that contain many enzymes and are responsible for regulating mRNA. For example, the P-bodies can decap the 5' end of mRNA, which exposes it to degradation by enzymes. It can also store the mRNA for later use or facilitate silencing by directing microRNA. You can see the P-bodies right here. Next, we'll discuss mitochondria, which you can see right here. Mitochondria contain the electron transport chain and are responsible for the production of ATP. Lysosomes are small organelles responsible for breaking down obsolete components of the cell. These include lipids, carbohydrates, and other waste. There are many lysosomal disorders, and I covered these in a section of the metabolism chapter, so we're not going to spend more time on these now. You can see the lysosome right here. Next, we'll discuss peroxisomes, which you can see right here. Peroxisomes are small organelles responsible for oxidizing very long chain and branched chain fatty acids. They also contain the enzyme catalase, which can degrade hydrogen peroxide. Peroxisomal diseases include Zellweger syndrome and X-linked adrenoleukodystrophy. The symptoms of Zellweger syndrome are not as important as knowing that it's caused by defective peroxisome function. This results in elevated levels of very long-chain fatty acids. In X-linked adrenoleukodystrophy, very long-chain fatty acids are unable to be properly transported to peroxisomes. In this case, the fatty acids accumulate in the brain and the adrenal glands, resulting in cognitive impairment and adrenal insufficiency. Proteasomes are tubular structures that degrade proteins marked by ubiquitin. Ubiquitin is incredibly high yield, so again, you need to know that proteins marked by ubiquitin will be sent to the proteasome and then degraded. Dysfunction of proteasomes can contribute to Parkinson's disease and Alzheimer's disease. In these neurodegenerative disorders, proteins are misfolded and should normally be degraded by the proteasome. However, the proteasome system is impaired, resulting in an accumulation of misfolded proteins. As the proteins accumulate, they can form aggregates in the brain and cause cell death. So, decreased proteasome activity results in increased misfolded proteins, which causes cell death. You can see the proteasome right here. Microfilaments, intermediate filaments, and microtubules are part of a system throughout the cell that's known as the cytoskeleton. Microfilaments are composed of actin polymers and are associated with muscle contraction, nutrient absorption, and maintenance of the cell structure. Intermediate filaments encompass many different molecules which are important for maintenance of the cell structure. Many of these molecules can be identified using immunohistochemical staining and can be helpful in identifying tumors. For example, keratin is an intermediate filament that is found in epithelial cells. So epithelial-derived cancers, such as squamous cell carcinoma, can be identified on histology by staining for keratin. Other intermediate filaments that you should be familiar with include desmin, vimentin, GFAP, and neurofilaments. Each of these correspond to a specific tissue and can be helpful in identifying various tumors that have been shown on the table. Microtubules are composed of alpha and beta tubulin polymers. These are important for cell motility, intracellular transportation, and mitosis. Molecules known as molecular motor proteins can transport cellular cargo on microtubule tracks throughout the cell. Dynein and kinesin are examples of these. Dynein proteins move cargo in toward the nucleus of the cell. So dynein dynes in. Kinesin proteins move cargo out away from the nucleus of the cell. Microtubules are a target for many drugs, so blocked by many drugs, which you can see in the table right here. These include chemotherapeutic agents such as vincristine and paclitaxel, as well as others. Dysfunction of microtubules can result in Cartagener's syndrome, which is also known as primary ciliary dyskinesia. Cartagener's syndrome is a disorder caused by a defective dynein arm. This results in immotile cilia. Let's draw this out. Cilia are composed of 9 pairs of microtubules, like this. If we were to zoom up on a pair of microtubules as if we were looking at it from the side, it'd look like this. So you can see that these represent the microtubules, and I'll draw in right here the dynein arm. Notice that the dynein arm connects the 2 microtubules. Normally, the dynein arm contracts and is responsible for allowing the cilia to beat back and forth. When the dynein arm is defective, the cilia are immotile. Because sperm and the fallopian tubes contain cilia, these patients will become infertile. Women are also at an increased risk of ectopic pregnancy because the oocyte is unable to properly move through the fallopian tube. Because the respiratory tree is lined with cilia, these patients will also have recurrent episodes of bronchiectasis. Finally, because the microtubules are necessary for the proper positioning of organs in utero, these patients will have situs inversus. This is a congenital condition that results in the reversal of organs. So, for example, the heart will be positioned towards the right side of the body rather than the left. Okay, you can see the cytoskeleton right here. Notice that this is only pointing towards one part of the cytoskeleton, but in reality it's shown all throughout the cell. Okay, let's do some questions. A 24-year-old female presents to the physician with concerns regarding breast milk production. She states that she has never been pregnant and has no children. She also notes that her breasts have enlarged over the past several months and she recently began producing milk. On exam, she is noted to have bitemporal hemianopsia. Her presentation is most consistent with the disorder caused by overproduction of a hormone produced by what cellular organelle? Hopefully from the question stem, you notice that this patient has galactorrhea and bitemporal hemianopsia. This is consistent with a prolactinoma. Prolactinomas are pituitary tumors that secrete prolactin. Prolactin is a peptide hormone, which means it's synthesized in the rough endoplasmic reticulum. So this hormone is produced by the rough endoplasmic reticulum. From the table, we can see that the rough endoplasmic reticulum is responsible for synthesizing peptide hormones. Examples of peptide hormones include insulin, ADH, and prolactin. You could imagine a similar question about the smooth endoplasmic reticulum. In this case, the hormone described would be a steroid hormone. Examples of steroid hormones include testosterone, cortisol, and estrogen. Okay, let's do another question. A 7-month-old boy is brought to the physician due to failure to thrive and cognitive delay. Upon further analysis, the boy is discovered to have impaired transportation of proteins to an organelle that is normally responsible for degrading obsolete components of the cell. What specific carbohydrate normally found on the proteins described above is most likely deficient in this individual? A key part of the question stem is right here. This patient has impaired transportation of proteins to an organelle that is normally responsible for degrading obsolete components of the cell. The organelle being described here is a lysosome. Because the Golgi apparatus normally sends certain proteins to the lysosomes, we can deduce that this is talking about impaired transportation of proteins from the Golgi apparatus to the lysosomes. Recall that I cell disease is a lysosomal storage disease caused by impaired transportation of proteins to the lysosomes. So we can deduce that this patient has I cell disease. Normally, mannose sugar molecules are phosphorylated within the Golgi apparatus, resulting in mannose 6-phosphate residues on proteins. Mannose 6-phosphate acts as a signal that tells the protein to go to the lysosome. In I cell disease, the phosphorylation of mannose is impaired. So, in answer to the question, the patient would most likely have a deficiency of mannose 6-phosphate residues on proteins. From the table, you can see that the Golgi apparatus is normally responsible for the phosphorylation of mannose on proteins. When this process is disrupted, the patient can develop I cell disease. Okay, let's do another question. A 12-year-old female presents with a non-tender, slowly enlarging mass near the left orbit. Biopsy of the mass reveals poorly differentiated cells that stain positive for desmin. What is the most likely origin of the cells seen on the biopsy? Hopefully from the question stem you notice that this patient has a tumor that stained positive for desmin. Recall that desmin is an intermediate filament present in muscle cells. So the answer is B. From the table you can see that desmin is found in muscle cells and can be associated with rhabdomyosarcoma, which is most likely what this patient has.